thank you very much for that introduction. And I'll be talking about stem cells in regenerative medicine. And this is just a cautionary statement about forward-looking statements. And as you all may be aware, there are two major hurdles in regenerative medicine, namely the problem of immune rejection and tissue shortage. And in addition to the shortage of whole organs such as heart and kidneys, there are literally hundreds of diseases caused by tissue loss or dysfunction. And with the advent of pluripotent stem cells, we now have a way to bypass, hopefully, both of those hurdles. So pluripotent stem cells are the body's master cells. They basically are immortal. They can grow forever. So, for instance, from a single cell, we've created an entire master cell bank that could literally treat everybody on the planet. And they're also pluripotent, which means that they can turn into virtually every cell type in the body. That includes insulin-producing cells, cardiac cells. Okay, great. And again, virtually every cell type in the body. So the first pluripotent stem cell applications are likely to be in the eye and the central nervous system. And the reason for that is twofold. These are immune-privileged sites. That means that the response to transplanted tissue is attenuated. And secondly, of course, there are major disorders that affect these organs. In the eye, vision loss costs literally up to $3 trillion worldwide. In the U.S., by 2050, it's expected to be over $700 billion for visual loss. In the central nervous system, neurodegenerative diseases are expected to cost over $1 trillion by 2030. And in the U.S., all sorts of other disorders such as multiple sclerosis, strokes, Parkinson, literally are going to cost the U.S. healthcare economy hundreds of billions of dollars. So at Akata, we're focused on the eye. The first application we're in trials with right now is for retinal pigment epithelium for macular degeneration, but we're also studying a number of other replacement cells, ganglion progenitor cells for glaucoma, corneal endothelium for corneal disease, mesenchymal stem cells for immune disorders, and also photoreceptor progenitors, which I'll talk a little bit about momentarily. So this is just a cartoon of the eye in the upper left. The light enters into the eye. It hits the photoreceptors. These are the cones and the rods that we see with. And underneath those are the layer of cells known as the RPE, or the retinal pigment epithelium. And they're the nurse cells. They basically maintain the health of the photoreceptors. So they transport oxygen. They recycle vitamin A and the photopigments, and literally dozens of other functions. And we've carried out the first two clinical trials in the United States, actually involving the use of retinal pigment epithelium derived from human embryonic stem cells. And we got approval from the FDA to treat two different diseases. One is dry AMD, and the other is Stargardt's macular dystrophy. And those are the leading causes of adult and juvenile blindness in the developed world, respectively. And so there's currently no treatment for either of those disorders. So this is a complicated slide, but basically what we're showing here is that these RPE, when they're transplanted into animals, actually can rescue the photoreceptor function and restore visual acuity to these animals. So in the upper left-hand column, you're actually seeing an increase in visual acuity in a dose-responsive way after transplantation of these of the RPE. And then in the lower right-hand corner, in those two greenish kind of figures, in the far right, basically you're seeing a cross-section of the eye. There are no photoreceptors left, no outer nucleated layer. And then the one just to the left of that, you're seeing very robust rescue of the photoreceptors, 5 to 7 layers thick. And based on that, we got approval from the FDA to proceed into clinical trials. We're carrying out currently 3 clinical trials, 2 in the United States, 1 in Europe. We've currently treated 38 patients. And we recently published some of the initial data on that, and we've seen no adverse safety concerns. This was primarily a safety study. And then the figure on the right in the middle, you can see a dotted circle. I don't have a pointer here, but basically that's the injection site. There's no pigmentation whatsoever. And then you can see with time the increase in pigmentation. And again, these are pigmented cells. And what we saw in these patients was an increase in visual acuity in the majority of the patients. and we didn't see that similar improvement in the fellow eyes that were untreated. And on the left, you can see a series of little white arrows, and you can actually see a nice monolayer of RPE that is attached to Bruch's membrane. And that actually, you can actually look into the eye in real time and actually see what's going on. So these are the results that we published. On the left, these are the results for AMD. And what you're seeing in the blue dotted line are the untreated eye, the fellow eye. And you can see at 6 months and a year, there was no improvement in visual acuity. So what we're doing is we're measuring visual acuity over the baseline. But in the dotted line, the eyes that were treated, there was about a 15-letter improvement in visual acuity. That's about 3 lines. We saw a similar improvement in the Stargardt's patient on the right. Again, about 3 lines improvement. But this doesn't really capture the improvement that's made in the quality of life of these patients. So these patients can now, read their computer, read their watch. They're able to go to the mall or the airport alone. One of the patients was a cowboy from Kansas who actually had 20/400 vision in one eye, legally blind. When we treated it a few months later, it was 20/40, and he could even ride his horses again. Just a couple weeks ago, we published another study. This was actually a confirmatory study that was actually done in South Korea. So this is the first pluripotent stem cell study carried out in Asian patients. In very similar results, we saw anywhere from a 9 to 19-letter improvement in 3 of the patients, and in the remaining 4th patient, just a 1-letter improvement, so it was basically stable. So again, importantly, it was a safety study, no teratomas, no ectopic tissue, no adverse events related to the transplanted tissue. But in addition to this RPE program, the advantage of using these pluripotent stem cells is you can turn them into every cell type in the body. So we've turned them into a number of different cells that are of importance in the eye. So for instance, we can turn these eye progenitors into retinal ganglion cells or into mature photoreceptors. So this is a study we just recently carried out where we actually were able to restore vision to completely blind animals. So in contrast to the RPE study where we're trying to prevent the progression of the disease, these are animals that are completely blind. They have absolutely no photoreceptors left. There's no outer nucleated layer after 3 weeks. So we waited till 10 to 12 weeks later, and we injected some of our photoreceptor progenitors into these animals. And then we looked to see what they might do in terms of function. So there were 2 types of tests that we can carry out. The first in the upper left is what's known as an optomotor test, where you can actually put a mouse into a drum that has these vertical bars that can rotate to the left or the right. And so you can test the visual acuity on either the transplanted eye or the untransplanted eye. And then on the lower, you can see a chamber. This is for light avoidance. So you put the mouse into the light chamber, and there's a little aperture, and you want to see whether he will try to avoid the light and go into the darkened aperture. And it turns out that 3 weeks after transplantation of these photoreceptor progenitors, a very significant improvement in both the optomotor response, that is the visual acuity, as well as in light avoidance behavior. And it was a very strong correlation with the improvement in visual acuity with the number of surviving transplanted cells.